Hi everyone, this is Mrs. Ching, and this is a video to help you learn about meiosis and to clarify the concept of diploid versus haploid. Okay, so first of all, I want you guys to understand that meiosis is not a cycle. It is happened once to make gametes. So here are your gametes, these four, right? That will be either your eggs or your sperm, or in the case of plants, can be pollens, right? So all of that stuff, so it happens that one time, then when they fertilize, the egg and the sperm fertilize becomes a full set of chromosome again, and through mitosis, then it becomes a full-grown individual. Okay, so let's go into detail about all of these things so that you can then understand this better. Okay? So I'm going to see if I can zoom in and help you understand this. Whoops. Okay, so here we go. In interphase, G1 is what we're doing here. And in G1, remember that it's 2N, meaning it's a diploid. And this should actually say 4 chromosomes. Okay? Because you have one that is, let me see if I can get the orange color up. Hold on a second. Okay, so here's orange. So you have one long orange and one short orange. And then you have these purple ones. So the long purple and the short purple. So if you were to pair these, the ones shown here, together, right? So if we were to take this one and put it over here, that's what it would look like. And so these are your homologous chromosomes. So this is homologous 1, this is homologous 2, second pair. So let's say you got your orange from your dad and then purple from your mom, right? That's what that means, homologous. Okay, so now we're going to zoom and look at what happens in interphase S. So in S phase, what happens in S phase, remember S stands for synthesis, so this is where DNA replication occurs. All right, so that's why it says, it should say 4 chromosome. But what happens is, notice that you have these centromeres. I'm going to color it in so you guys can see it. These are for 4 centromeres. So when you duplicate it, you're making sister chromatids, right? So these are your, oh sorry, should have done it with a different marker. Here, this size. Okay, so these are sisters chromatids because they're identical. So that's why they're joined at the centromere. Okay, and then you count one, if there's a centromere, you count the number of centromere. So that's why there's 4 chromosomes. 4 chromosomes here. Okay, chromosome number hasn't changed, even though you've duplicated. Okay. All right, so then what we're going to do now is go to metaphase. Oh, and remember prophase is when crossing over occurs. This is your tetrad over here. So here's your tetrad. All right, so now we're going to move on and go to metaphase. So metaphase, what happens is each of these are homologous chromosomes. then they are going to line up perpendicular to the spindle fibers. But notice that they are stacked up on top of each other, right? So that when you get to anaphase, the homologous chromosomes separate. Let me actually erase this one. Oh, it erased all of it. So hold on, let me redraw that. I just don't want to confuse you. So each one of this is homologous chromosome. Okay? And that's what splits in anaphase 1. When you get to telophase, notice how many chromosomes you have. You're only gonna, how many centrioles do you see here, right? So you only see 1, 2 in each cell. So that's why that's 2C. Okay, I don't know what that is. Don't worry about it. So this is a haploid. That's why it's N. Okay, so this is another haploid. Also another N. Okay? So then when you get to prophase here, each of the cell, prophase 2, each of the cell will be 2 chromosome and a haploid. So haploid here, because you divide it, 2 centromeres. Okay, metaphase. The thing that you need to remember here is that these are chromatids that are being separated. Okay, so in anaphase, the chromatids separate. Okay, same thing with this cell, chromatids separate. So when the chromatids separate, you have now 2 chromosomes in each. Okay, so again, this is not right. This should be 2 chromosomes and okay, in each cell. So let's look at our gametes that we made. Okay, so in cytokinesis, each of these have 2 chromosomes and an N. 2 chromosome and an N. There's no centrioles here, but you count this whole thing as 1. Well, let me redraw that better. Okay, so this whole thing is 1 chromosome. Here's your second chromosome. Okay? So imagine what could happen is that, imagine, let's say these are sperm, and then you get an egg that has something that looks like this. Hold on, let me get the colors back up. Okay, so you get another that looks like this. Maybe I'll make that one a different color. Make that a different color. Okay, so let's say this is the egg, and these are the sperm. And this joins with this. The resulting zygote is going to look like this, where you have 1 long blue chromosome, right? And then you're going to have one that's going to have the bottom tip is a little bit orange, and then the rest, or purple I mean, and the rest are orange. Okay. So, and then like this. And then this would be the homologous pair. Oops. Okay, so once it's joined, this would be then a fertilized egg. Which is your zygote. This word right here. And it has 2 N again. Okay, I hope that helps. Come talk to me if this still doesn't make sense. I'll be around next week.